I'm Carlos Chacón. I'm a PhD student at the Spanish National Cancer Research Center. And today I'm going to show you who we are, how we have adopted Binder design tool settings, and the tool that we have constructed with BinderFlow, the pipeline that I've built with all the tools that are available right now. So first of all, we are two structural biology groups from the Spanish National Cancer Research Center, the latest lab and the Yorkat lab, and we work mostly with cryo-EM data processing. When the Rosetta Efficient paper came out, we started interested in protein design, and more specifically in binder design, so we incorporated to our pipelines. We have a computing cluster with approximately 15 nodes on 40 consumer GPUs. Cryo-EM is a very computationally intensive activity. So we started doing protein design following the pipeline described by Watson et al. In the Rosetta Efficient paper, as it's generating backbones using Rosetta diffusion, assigning end sequences using protein MPNL, and discovering those designs using AlphaFold 2 initial guess, where n usually represent tens of thousands of designs. The problem is that when we run this pipeline in our setting, we had some problems, including that these are very large jobs that occupy a node for long periods of times, hours to days, and this in combination to cryo-EM data processing led to cluster clogging. The other problem was that you had to manually handle all the inputs and outputs of each step, which can be a source of error and also limits the accessibility of the pipeline to people that are not very familiar with computers and not very proficient with computers. And also in our settings, it led to misuse of the computational resources. It's very hard to know a campaign success rate beforehand. It was very often that you end up with more designs that the one can be screened, or that you spent a lot of time in a campaign and no good results were yielded because it was not a surface that was sensible for binder design. Finally, the parallelization of the process was not straightforward, and thus this further increased the HPC clogging. So we needed a pipeline that split the design campaign into small batches that lasted from minutes to a few hours. We need to reduce human intervention to a minimum, end-to-end pipeline. We will also need to use a computational resources intelligent, so not overuse them or infreuse them. And we will also need a pipeline that parallelizes the process across nodes and GPUs. Our solution to that was Binder. With respect to the workflow itself, BinderFlow works just as the linear pipeline, so backbone design with RFT, protein MPNN, sequence assignment with protein MPNN, and in silico scoring with AlphaFold 2. But we added two steps in order to improve efficiency. Between the RFT and the protein MPNN steps, we added automatic filtering of backbones, which in our hands are not compatible with expression, like single helices or hairpins. And to do this filtering, we use DSSP. And we also added a second scoring step using Rosetta, like others have done, for example, in Minecraft. And we added other metrics like KPSI. The most important addition of BinderFlow is with respect to how it's the binder design campaign organized. So this time, instead of having very large jobs, you divide the campaign into small batches that run end-to-end. In these batches, the n is tens of thousands of designs represent tens of designs. So these small batches can be run in timescales of minutes or hours, depending on the length of your target, your binder. It also automates the inputs and outputs between steps. You don't have to manually handle all these results. And we use silent files in order to keep everything nice and tidy. And also it parallelizes the campaign between nodes and across GPUs in order to improve real-time efficiency. The final advantage of BinderFlow is that it allows for live monitoring of the process. So you can stop a campaign early in case you are not having any good results, or the pipeline can stop automatically once you have enough results. We found out that this was very useful in our settings, at least because we could send the jobs, and these small jobs will enter when a node decided from these cryo-EM data processing jobs, which are very computationally intense processes. Another advantage of the live monitoring of the process is that it can be easily visualized, and to that end, we created a Python web app, which is called BFmonitor. BFmonitor allows you to monitor how the campaign design is going. It eases the task of debugging the pipeline if something is off, and also allows you to extract not only the PDBs, but also the complementary DNA sequences ready for work. The first step will be the live watcher, which is a step in which you can follow live depiction of the campaign using the scatter plot. You can select which in silico thresholds to use in order to consider a design and in silico success, what we call a hit. And also you can make previous comparison between designs across more important metrics. As you can see, there are different colors in the scatter plot, and it is because only those designs that pass all thresholds are colored according to its length, and the ones that do not pass thresholds are colored gray. The next step will be the pipeline tracking, which is useful to debug the process. You can track the state of each batch, whether if it's the Rosetta Fold diffusion, the filtering, the sequence, scoring step, and you can see whether if a batch has failed or completely, or has finished successfully, and you can inspect the logs of the step. And the final tab will be the extraction tab, in which you can visualize the structures of each of the hits as according to the threshold defined earlier. You can select them to extract the PDBs from the silent files that we are using, or you can also extract the complementary DNA sequence using column transform. I must say that right now, instead of using column transformer, we're using John Berko from the SAP DMLX GitHub, and we're working to implement it also in the BFmonitor. So, respect to how to install BinderFlow, you have to first install using Conda the different tools needed for the listening project from the GitHub repositories. The only one that you don't have to install from the respective GitHub repositories is the DLB binder design that you have to use the fork version, which extracts the matrices from AlphaFold to initial guess. Then you have to clone the BinderFlow repo and run the bash script install BinderFlow.sh. This script creates an environment that is needed for the filtering step and for the BFmonitor. And finally, you have to install the SSSP for the filtering step. Then, lastly, you have to modify the config.sh file according to your system settings. In this file, which is inside the binder, you have to specify which are your models path, the names of the models environments, and also your personalized configuration. BinderFlow runs as a background job, which is this BinderFlow.sh, which detects how many jobs are running, and in case one job finishes, sends another batch, or if enough hits have been already designed, it stops the campaign automatically. The settings of the campaign are defined in a JSON file, which looks like this, in which you have to define the number of nodes that you want to use in parallel, target input, the total number of hits, among other things. In this JSON file, you have to also define the design strategy. This is because BinderFlow is right now compatible with 3 different design strategies. The initial binder generation is described by Watson in the R paper, so RFP and focus initially. It is also compatible with partial diffusion, in which starting from a validated backbone, you slightly tweak the structure in order to improve the in silico metrics and hopefully the in vitro metrics. And then also it's compatible with sequence diversity, which is how we call this design strategy, in which starting from a validated backbone, you skip the RFD step and generate thousands of sequences trying to improve the metrics. So once you have sent a job and it's been running for a while, you will end up with a directory looking like this. And the most important files and folders are the output folder, inside which is each of the batches into which the campaign is splitted, which I call run K. Then it is also very important CSV, which is a CSV file where all the metrics for each design are stored. And also this hidden folder, which is called Chacón, which is a folder that stores the alpha matrices as they are computed in order to reduce prediction time. Finally, when you run BFmonitor.py, which is the monitor tool, it will create this hits folder, inside which you can see the PDBs, the FASTA sequences, and the DNA sequences from the extraction tab. And also, once enough hits have been generated, this empty file called campaign done will be created, pointing out to the background job that enough hits have been designed, and thus that it has to stop. If we go into a little bit more detail into how the batches look like, inside one batch, we will have this structure in which we will have some PDBs and TRP files, which is the output from RFD. The substituted marks the ones that pass the secondary structure filter. Then we will have a list of silent files, which are the input and outputs from Protein MPN and AlphaFold2. And we will also have this series of JSON files, which contain the pi information for each prediction. Respect to how we name the designs, we are using this nomenclature, rank K, GPU I, design N, substituted DLT design E, where K is the batch number, I the GPU identifier, N is the design number, and M is the sequence number. And once a batch has finished, it will create this empty file, which is called rank K down, will indicate to the background job that the batch is completed and will send another one. So, as a summary, BinderFlow is an automated pipeline that divides a binder design campaign into smaller batches, facilitating the coexistence of protein design with other computational intensive process. Moreover, this batch-based organization is compatible with the parallelization of the campaigns, improving real-time efficiency, and also it uses efficiently the resources available. respect to the future perspectives, we'd like to improve the installation of the different models. Probably containerization like Roscoe does is a great option, or the IRC run that Hope mentioned here. We would also like to adopt new tools like Roscoe Diffusion 3 and Legon and PNF. Also, we would like to add new strategies like unconditional monomer generation or symmetric oligomers, since right now BinderFlow is only compatible with binder design. We would like to improve the adaptation of the workflow to HPC clusters. Then we probably should remove the pipeline job and the config.sh file. And finally, we'd like to improve the accessibility. Then 10, we have a master student called Diego working in another tab for the BFmonitor, which is called the input tab, in which you can create a new campaign, load a PDB, crop the structure, select different hotspots, and select the different settings for your run, and send it from there. This is very useful because this way we reduce the use of the terminal to a minimum and improve the accessibility of Binder design to people that are more focused on the wet lab part, but are not very proficient or do not have a lot of computational skills. So I would like to thank both the Leiros Lab and the Yorkas Lab, especially Ed and Diego, who have been working also in this project, and also the people in the lab that have been working with putting binders like Maria Moreno. So thank you all for attending, and I will be happy to answer your questions.
Great, thank you very much. Great talk. Again, if you have questions, paste them in the Zoom talk. And I'll start with some questions, but actually I'll start with the comment first. I think implementing containers in BinderFlow and actually in AI.zymes is super easy already directly, because all you have to do is change the command line to have obtainer run whatever, and then you can use the container directly. So that step should be pretty straightforward, and it does make it much nicer for the end users, because then you can just do a few Docker pulls or obtainer installs.
Yeah, I agree. Like, we haven't done it with containers because I think I'm at the same a little bit old school, and I work with Conda, and I think I'm used to, but I should work to containers. One gap that I have is that for people, like, it's okay to have like a discussion here. Like for people from, for example, from web labs that are not really used to computer handling and all that stuff, will container make things easier to install? I'm not familiar with containers, so it's a genuine question.
It will, because it's basically just downloading one file that sort of works independent of everything else. So there's less chance for things go wrong. Conda is super finicky, right? And nowadays it takes forever. Even if you're using Mamba, it still takes forever to resolve the dependency and stuff like that. So I think, containers are the way to go. And as I thought, it's really just like a one line change in your current setup. Happy to talk more about this offline, but it should be pretty cool. Then I'll start with some technical questions. So I guess you're doing the same as we all are. So you're writing the input files in Python and then running the tool and then parsing the input files again, right? For example, for RF diffusion or like protein MPNN or stuff like that.
And so can you repeat the question?
Yep. So technically the workflow, if you want to run RF diffusion, you basically make BinderFlow write out a YAML file, and then you pass this to RF diffusion, and then you read it, the PTPs, right?
Yeah, we have this. Then from the JSON file, we put all the input to a SH, to a bus file, and this bus file is the one that runs in Fusion and Spy 3 with all the stuff.
And then how are you saving? Because you might want to pass some information from, I don't know, the RF diffusion part later to protein MPNN. Do you keep track of this information in a POST file or in a CVS file or
information from RFD to other steps?
Yeah, but maybe for few binder design, you don't even have to do that, right?
Yeah, I think that at least we didn't have the need to pass information from RFD apart from the PDBs itself.
Yeah, because like in Proscope, when we do motif scaffolding, we have to pass the part that's newly generated so that only this part gets redesigned with protein MPNN and not everything.
Yeah, we have a flag, which is called residues. And this residues flag in the JSON file, you can specify which residues to give, not only in Rosetta diffusion, which just start from GFQ to motif scaffolding, but also in protein MPNN.
Very cool. How well is your code structured in a sense of how easy it is to add AlphaFold 3 and Boltz Gen and stuff like that?
I think it's not the plugins from all. at this, it shouldn't be very hard, but I think you have to get familiar with the code. So it has some work getting familiar with the code and how it's written. The way we work is that we have a master script which is BinderFlow datasets, which call another script, and then that script call the different tools that we're going to use. And so a lot of if and else conditions to go to one design part or to another design part. So in order to add these new tools, for example, AlphaFold 3, we have added in this input tab for triggers validation only with the hits. And it was not very hard, but I wouldn't say that it's, like I said, like the plugins from our.
Very nice. And especially like the live monitoring idea. It's very nice. Are there any other questions? Well, I think this is all still from science. I think if there are no more questions, maybe like one more. How open are you to pull requests? And
yeah, we're very open to any, if someone wants to contribute to the GitHub, I forgot to put the link, which is something that always happens to me. But this is on GitHub and you can download it. If you want to contribute, we're very happy to work with you. And also, if you need help installing the things, since, as I say, Conda is a little bit a nightmare sometimes, we're happy to help.
then thank you again for a great talk.